Welcome. In this session, I'm going to discuss about translation. And before I start discussing about it, I would like to remind you about the central dogma of molecular biology. So the DNA undergoes replication by itself and helps in formation of copies of its own, and this DNA can form RNA by a process of transcription. So here not just mRNA formation from DNA is called transcription. So even tRNA formation, okay, rRNA formation or any other type of RNA formation from DNA is called transcription. Now this mRNA will be translated to form protein, right? So already we have seen the replication of DNA, we have understood the process of transcription, right? And now the last part of this, the central dogma of molecular biology, that is translation. So now let's look at the process of translation, right? So we'll see the definition first. Translation is a process of polymerization. So it is a process of polymerization of amino acids, right? So basing on the information present on the mRNA, the amino acids will be arranged according to that information. That's what we call polymerization of amino acids to form a polypeptide. So that results in the formation of polypeptide. So here, say suppose this is a valine amino acid, then next methionine, then glutamic acid, and then valine, okay, and then methionine. You can see there is attachment of amino acids, and this is what I call it's going on adding. So it's called polymerization of amino acids. It's called polymerization of amino acids, and this results in the formation of a polypeptide chain. So this all will become a polypeptide chain, right? And of course, that is what we call the gene expression. So where we have seen the DNA transcribing mRNA molecule, and that mRNA undergoing translation and finally forming a polypeptide that will show its effect, and that what we call it expression of gene. Now let's look at how this polymerization of amino acids to a polypeptide takes place. The process of translation requires, so what are those requirements for this translation? One, right, require RNA molecules. So RNAs, so all that mRNA molecule, then rRNA molecules, and even the tRNA molecules are required, right? And in this case of rRNA molecule, so this will be the structural component of ribosome, and the next one are ribosomes. Of course, all this part, the transcription, translation, we are going to study in case of bacteria, that is E. coli. It means in prokaryotes, we will understand first. So later, we will look into eukaryotic organisms. That is complex, but okay, our main theme, whatever that transcription, translation, we are discussing only in case of E. coli uh, bacteria. So ribosomes, then the requirements RNAs, ribosomes, and then amino acids, and then enzymes are the requirements for the process of translation. And we know that this mRNA molecule can be monocistronic or polycistronic. So as I told, we are going to discuss mostly in case of bacteria. It will be right polycistronic. And tRNA molecules already we have seen in the last session, right? The actual structure of the tRNA will look like inverted L, and but in the clover leaf model, the tRNA will have total 4 arms. So it has 4 arms, right? And this is the 5 dash, that is 5PN, and this will be third OH end. And I told you that this is the point where amino acid is going to bind. So amino acid acceptor end is called amino acid acceptor end, or binding site you can call it. And opposite we have anticodon, right? And this is a codon recognition site. So that will have anticodon here. Okay, and I told you that this is stem and this is loop, and these two together form arm. And in some cases we will find a arm variable arm. The stem shows pairing, whereas loop will not show pairing. So there will be nitrogen bases that are not paired, but in case of stem there will be pairing. So you can see like this, and here we have CCA is the code that usually seen on the third OH end. Okay, and just write this side, the stem, loop. So these two together form arm, right? So here you will see like anticodon. So this is anticodon, so which will be paired with the codon present on mRNA molecule. So this is how the tRNA molecule looks like, right? So this tRNA molecule is going to have two functions. One, going to read the information on mRNA, and then according to that it's going to bring the amino acid. So on this third OH end, it's going to carry the amino acid, right? And the type of amino acid attached to this tRNA, it all depends on the codon on mRNA, right? Now we'll also see the ribosomes, right? So the ribosome in prokaryotes will have 3 subunits, okay, 50S and 30S. So this is the larger subunit, and this is smaller subunit, larger subunit and smaller subunit. Okay, and of course when there is no process of translation taking place, these two subunits will be separate, and they will come together only when there is the process of translation, right? Now inside the ribosome, especially in the 50S, So in the 50s, it will be showing, right, mainly three regions. Of course, in the textbook they have mentioned, okay, two. Here I'm going to mention, so let's say suppose this is the 50s, it will have three regions. So that's E site, P site, and A site. So in the textbook they have mentioned only about the P and A sites. Okay, so here the E actually stands for the empty site. This is the position where it will be occupied by tRNA that already delivered its amino acid. So for that reason, they might not mention about this empty site. Okay, then P site, which stands for peptidyl site. So because it is at this site, there will be a formation of band. So when bond, when two amino acids are present in P site and A site, the bond formation between the amino acid takes place. That I'll shortly explain. So here P stands for peptidyl site. It stands for peptidyl site, and A stands for amino acyl site. Right, so these are three sites that are seen in larger subunit of ribosomes in case of prokaryotes. Okay, of course, the same will be seen in case of eukaryotes also. Right, the P and A sites, these two are very important. When two tRNA molecules which are charged and are present side by side, then the bond formation between the two amino acids takes place, right? And during the process of translation, the 50s will be, okay, binding with the 30s. So all this 30s, so this is 30s and this is 50s. Okay, so both of them, they bind, right, to form the complete ribosome. And we know that the ribosome is composed of rRNAs plus some 80 different proteins. So just proteins and rRNAs make up a ribosome, right? So now we have seen the requirements of this translation process. Now coming to this translation, it takes place in, okay, three steps. One, there is initiation. So I can say chain initiation. The next one, elongation. Chain elongation. So that is polypeptide chain elongation. And then lastly, termination will be termination. Okay. So here the initiation begins with, of course, the codon AUG and ends with, that is termination, ends with the codons, that is stop codons. So on the mRNA, the initiation will be with the AUG and termination will be with the stop codons. Okay, so now let's look at this process of initiation. So here in the initiation process, there are two steps that take place. One, it's called activation of amino acid. So step one in this case is activation of amino acids. Then coming to step 2, after activation of amino acids, the next one will be, okay, that activated amino acids link to tRNA molecules. So that is, okay, so linkage to tRNA molecules. So activated amino acids will be linked to tRNA molecules. So now let's look at the first one, how the activation of amino acids takes place. So step 1, activation of amino acids. So in this process, right, the amino acid is going to react with the ATP molecule. We know the ATP is the source of energy, and this takes place in the presence of an enzyme called amino acyl tRNA synthetase. So it takes place in the presence of enzyme amino acyl tRNA synthetase, and also in the presence of ions, that is magnesium ions are required for this process. So amino acid will react with the ATP, which is a source of energy, and this results in the formation of amino acyl AMP, right? So we know this ATP, when the phosphates are removed, so when two phosphates are removed, it will become AMP. So that is, I'll write 2pi. Right now, this is charged amino acid, amino acyl AMP. Now, in the second step, okay, so charged amino acids linked to tRNA molecules, right? Of course, we know that each tRNA molecule is specific to an amino acid. So it is not like out of 20 amino acids, each amino acid has, of course, its own tRNA molecule. It's not like one tRNA molecule carries all the amino acids. That is not the case. So here charged amino acids linked to tRNA molecule. So that is amino acyl AMP. So amino acyl AMP. So this is a charged amino acid. Charged amino acid. So which is going to, right, link with the tRNA molecule. So amino acyl AMP plus, okay, tRNA molecule, again in the presence of enzyme called amino acyl tRNA synthetase. Amino acyl tRNA synthetase, and again in the presence of magnesium, okay, ions, it's going to form amino acyl tRNA plus AMP. Okay, so plus AMP. So right, so AMP is going to get separated, and this amino acyl is going to combine with the tRNA molecule, right? So now the tRNA molecules, okay, are charged, and now they are ready to carry these amino acids to the site of uh, you know protein synthesis. So these are two steps in the process of initiation. So now let's look at the process of elongation, right? Of course, for all this, there will be several factors required that we will see it later. Okay, so here the mRNA molecule, so 5' to 3', okay, and on this you will find, okay, sequences and so and so, and say here is UAA. So this is the stop codon, and you can see here the beginning is the start codon, and this is mRNA molecule, right? So now the process of this begins, that is translation begins when smaller subunit, okay, comes in contact with the uh, mRNA molecule, right? So I think already I have mentioned inside the cell. Okay, so here there will be amino acids in the cytoplasm, and this is the mRNA molecule that is being released. So in case of prokaryotes, the site of transcription and translation is same because there is no compartmentalization. Okay, and in cases the translation can begin even before the transcription is completed. So say suppose here this is the DNA molecule, and still the mRNA is forming here. So you can see mRNA molecule forming here, and the process of, so this is mRNA, and so this is DNA molecule. Okay, and the process of ribosome. So this is ribosome binding it. You can see there is still transcription going on even before that is finished. Okay, in case of I'm telling prokaryotes. So even before transcription is completed, translation can begin. But this won't be in case of eukaryotes. So that won't be in case of eukaryotes. Only after the transcription is completed, because we know in case of eukaryotes, the mRNA will have some additions. So which I told in case of eukaryotic mRNA, which will be monocistronic. So it will have additions like methyl guanosine triphosphate. So this is called capping, which will be seen on 5 dash, and there will be a poly A tail. So this is called poly A tail on, okay, third end. So this is third end. Let's mention, okay. So on the third end there is poly A tail, and this is actually called a tail here. So it will have tailing and capping, right? And after that you will be finding some region that will not be translated, so it's called 5 UTR. And here also you will see on both sides third UTR. Of course the same you will also see in case of prokaryotes, but there is no capping and no tailing. So this is in case of eukaryotic mRNA, which I have discussed earlier, and this is mRNA in prokaryotes. mRNA in prokaryotes. Of course, there will be region, so 5 dash. So this region is 5 dash UTR, so that will be not translated. And after this also, we will be having so 3 dash UTR. So there will be 3 dash UTR, so that will be not translated. And of course in case of mRNA in prokaryotes, there is no capping and no tailing. So because of this additional features, and moreover this whole process takes place in nucleus. Only after this completion it will be transported to, okay, out of the nucleus, where the process of translation begins in case of eukaryotes. So such condition is not seen in case of prokaryotes. Right. So I'll just mention, so in prokaryotes, transcription and translation occur at same site or location. So that will be the cytoplasm of the cell. Right. So but in case of eukaryotes, transcription okay, occurs inside the nucleus. So it occurs inside the nucleus, whereas translation occurs outside the nucleus. So it occurs outside the nucleus. That will be the cytoplasm of the cell. So that's a difference between the prokaryotes and eukaryotes about transcription and translation. Of course, there are many other differences, right? So keep this important, as it's important. So here this is the DNA and the mRNA. Even before mRNA is released from DNA, the process of translation can begin in case of prokaryotes. So now the translation begins when the smaller subunit comes up here, and then so it's bind over here. So this is uh, 30s, smaller subunit, and then we will see attachment of larger subunit. We'll see the attachment of larger subunit. Okay, right. So this larger subunit, so it will be right. How it will be? So this is 50s and this is 30s. Okay, so it arranges in such a way that this will be the first site. Okay, so here is the P site, and here it will be the okay A site, CCU. And now the tRNA molecules will come over here, and this will be having UAC. So only the one with the anticodon UAC will be able to bind here, and this will be carrying the amino acid methionine. The next one, the next tRNA will be carrying okay GGA. So it carries anticodon GGA. So the one which carries this will be carrying proline. So this CCU, we know that according to the genetic code, it codes for proline. Okay, so this is methionine, the initiator amino acid. Okay, so methionine, and then okay, proline. Uh, so this is anticodon, and this is codon part. So when two charged tRNA molecules are present side by side, so what happens is there will be a bond formation between these two, right? And here, of course, the ribosome will be having that catalyzing unit. So that's ribosome also acts as catalyst. So it also acts as catalyst. So how it acts as a catalyst? It contains rRNA. So that is 23S rRNA. So we know that already ribosomes will be having rRNAs. Already I have mentioned in earlier sessions about the ribosome and components of rRNA molecules present in larger and smaller subunits. So this ribosome will be acting as catalyst, and it will be having 23S rRNA. Okay, and this is what we call enzyme that acts as a catalyst, and it's called a ribozyme. Okay, it's called ribozyme. So here the ribozyme is 23S rRNA molecule which help in the formation of peptide bond. So help in formation of peptide bond, of course, in bacteria. So that will be in bacteria, right? Right, so that catalyst, so the ribosome acts as a catalyst, and this is what we call the 23S rRNA, which is a component of larger subunit, and this is going to form the peptide bond. So here is the peptide bond, and the methionine attaches over here, and this is removed. Right, now once this is finished, the ribosome is going to move in this direction. That will be from 5 dash, okay, towards the 3 dash. It's going to read in this direction, so that is 5 dash to 3 dash. Now once this is finished, the larger subunit is going to move one codon right. It's going to move one codon right. So now, so it has moved one codon right, and that will be followed by the smaller subunit. Will be followed by the smaller subunit. So 30S, and this is 50S. Now what happened? So earlier this was in P side and this was in A side. Now what happens is, okay, this one comes to the P side. Now A side is ready to receive another tRNA molecule. So there is incoming of the tRNA molecule, and now this is a tRNA molecule that already delivered its amino acid, and it can be moving out, right? So now, right, so into this another tRNA molecule comes over here, and this will have anticodon UGU. So it will have UGU, and this codon ACA will be coding for what? It will be coding for 309. So it will be coding for amino acid 309. Now, so these two charged tRNA molecules, when they are present, now this entire, okay, the two amino acids are going to form a bond with this 309. Okay, so here peptide bond, and this will be proline, and peptide bond, and this is methionine. So this entire, okay, will be updated over here, right? So now as this ribosome moves on the mRNA towards the right, you can see there is increase in the length of the polypeptide chain. So now once this is finished, so you can see here this will be moving out. Okay, so I'm going to remove this. So it has moved out here, and now again the ribosome moves towards right one codon right. One codon right, and that will be followed by the smaller subunit. And say suppose we have A, okay, C, A, C, some sequence, right? And now this is the P side, and now it will become the A side. So now another tRNA molecule that matches with this codon will come over here. And this will have, okay, the anticodon AGU will have anticodon AGU. Now this one, the UCA, will be coding for amino acid, right, serine. The UCA will be coding for amino acid serine. So once these two are present, now this entire chain comes over here. Okay, so here it is 309, then proline, and then methionine. So you can see there is elongation of polypeptide chain. So this is the polypeptide chain, and this proceeds till it comes to a stop codon. So any of the stop codon, okay, UAA, or you can take UGA, or okay, UAG, all these are stop codons. If any of these stop codon is present, okay, and we know that for these stop codons there are no tRNA molecules, and that results in, okay, chain termination. So that is the last part of this translation is chain termination. Once that is finished, now this polypeptide chain will be released into the cytoplasm, right? So okay, so that's the whole process of translation. Of course, one mRNA can be read by many ribosomes simultaneously. So you can see here mRNA. So in this case, one ribosome have come up here, and you can see this is already having a lot of amino acids, right? So this is a polypeptide chain, and you can see that is being followed by another one, okay, and this is another one, okay, and moving in this direction, that is towards the right side, okay, right? And reading of ribosomes means many ribosomes at a time. So we call, I think in the first year we studied this, it's called polyribosome. So this is, right, right, so polyribosome where many ribosomes reading mRNA simultaneously. So many ribosomes reading mRNA simultaneously is what we call polyribosome. Because there are other words like can also use polysome, okay, or ergosome, or some other words that we'll see it later. So that's all about the process of translation. Right, so before I end up this session, just one more point I would like to add. So here, for initiation and termination, for these two processes, okay, factors are required. So there are some factors required. So we call initiation factors. So there are certain initiation factors, and there will be, right, termination factors, okay, or I call releasing factors, right? So I say, or release factors, or again initiation. For initiation process, it requires initiation factors, and for termination it requires, okay, release factors. So once that here the ribosome comes over here, right, so what happens is the releasing factors will help in releasing this polypeptide chain, okay, out from the ribosome. Fine. Right, so that's all about the process of translation. So in the next session, we will see about regulation of gene expression.